Hi there. In this video, we are going to discuss the third hallmark of the carcinogenesis, which is altered cellular metabolism. In addition to other characteristics, cancer cells also show a variable or changed way of their cellular metabolism. What they do, even if there is abundant supply of oxygen, they still move towards the aerobic glycolysis, or they rely upon the glycolysis for their metabolic demands. We know that glycolysis cells rely on glycolysis when there is not sufficient oxygen present. When there is deficiency of oxygen, then they rely on glycolysis or fermentation. However, the cancer cell, in the presence of abundant of oxygen, still rely on glycolysis, and this phenomenon is termed as Warburg effect, or it is also known as aerobic glycolysis because it is happening in the presence of oxygen. So in the cancer cell, the Krebs cycles and the you can say oxidative phosphorylation is negligible, or you can say at a very low amount. The principal metabolism going on in them is glycolysis, and this glycolysis is monitored usually by using fluorodeoxyglucose, which is a labeled glucose molecule, and it is monitored by positron emission tomography scan, and most of the cancer cells show this scan positive. It means that they show increased glucose uptake. So cancer cells usually have an increased demand of glucose and increased glucose uptake and increased fermentation or glycolysis of the glucose, and they convert glucose to lactate and so on. However, they don't shunt the glucose to the Krebs cycle much. And this is also this Warburg effect is not limited just to cancer cell, but it also happens in the rapidly dividing cells of the body, for example embryonic tissues, as well as the lymphocyte during the active infection. So why it happens? Glycolysis only yields 2 ATP. However, in case of Krebs cycle and oxidative phosphorylation, 36 ATPs are formed. However, when cells want a deficit of the ATPs and choose to be a pathway in which the production of ATP is very low, it is because the rapidly dividing cell, including tumors, including embryonic tissue, including lymphocyte, they also require the metabolites to synthesize their own products. For example, they need metabolites to synthesize lipids, they need metabolites to synthesize proteins, as well as DNA, as well as RNA. So there are metabolic needs. They don't require much of the ATP when they will not be having these metabolites. So they don't use all of the glucose for the synthesis of ATP. Instead, they divert this synthesis of ATP towards these metabolites so that they can grow and they can divide and produce more and more cells. Okay, so this is the reason why they don't shunt their all of the glucose to the mitochondria and for Krebs cycle. However, some shunting occurs, and that is in such a way that it only yields about 4 ATPs, and the products, metabolic products from the Krebs cycle are also used to synthesize their metabolites. For example, they use acetyl-CoA from the mitochondria in the synthesis of lipids. So in cancer cells and in rapidly dividing cells, the glucose is oxidized to the pyruvate and in the glycolytic pathway to yield 2 ATP, as well as multiple you can say metabolites which are used to synthesize their own chemicals which are necessary for their division. And there is also a very small amount of shunting to the Krebs cycle and mitochondria, which yields about 4 ATP, and the some products, the metabolites from the Krebs cycle, are also used to synthesize these metabolic products for the division of the cells. So this is phenomenon is known as Warburg effect. So in normal cells, for example in tumor, so in normal cell, for example in embryonic cell and in lymphocytes, this process of Warburg effect is regulated. Okay, cells rely on aerobic glycolysis only at the time of need. When need is over, this process shut down. However, in tumor cells, there is no proper regulation, and this Warburg effect continues. Usually, tumor suppressor genes oppose the Warburg effect. However, we know that in cancer cell, tumor suppressor genes are mutated. So when there is a mutation, there will be no opposition to Warburg effect, and tumor cell continuously rely on this pathway and continue to divide. Moreover, there is how this pathway basically works. The receptor cells, which are the growth receptors, we know that in the cell membrane there are growth receptors which lead to the downward signaling. This receptor, when a growth factor binds here on a, supposedly this is cell membrane here and this is a receptor here, and when a growth factor binds here, in addition to leading to downward signaling towards the nucleus, this receptor also upregulates the intake of glucose into the cell. It upregulates glucose intake as well as it upregulates the glycolytic enzymes. Okay, so these receptors basically play a role in Warburg effect. When growth factor continuously stimulate in the receptor, or there is some mutation due to which receptors are continuously being stimulated, they lead to the increased uptake of glucose as well as upregulation of glycolytic enzyme, and they also inhibit the last pathway of the glycolytic pathway, which leads to the phosphoryl pyruvate conversion to the pyruvate. However, the other major factors also play a role in this pathway. Moreover, we previously discussed that RAS mutation, RAS mutation, is also involved in carcinogenesis, and this RAS mutation is also involved in the Warburg effect. How it plays a role in Warburg effect? It also leads to the upregulation of the glucose intake, as well as it leads to the shunting of this shunting, in which acetyl CoA and metabolic intermediate from the mitochondria shunted to the synthesis of lipids. So this RAS mutation is also leading to the Warburg effect. Moreover, there is MYC, which was our last product in the previous lecture. We studied that it also promotes growth, and it also lead to the upregulation of glucose and upregulation of the glycolytic enzyme and shunting of the mitochondrial intermediates to the synthesis of cellular metabolites. So RAS mutation receptors, RAS mutation as well as MYC, all these promote to the upregulation of glycolysis and shunting of the metabolic intermediate to the synthesis of lipids, proteins, and cellular components. So they contribute to the Warburg effect. And tumor suppressor gene basically oppose this Warburg effect, and the defects in tumor suppressor gene, including NF1, which was involved in neurofibromatosis, and PTEN, which was involved in certain leukemias, and TP53, which was involved in multiple kind of carcinoma. These tumor suppressor gene oppose this Warburg effect, a continuous Warburg effect in the cell. However, when they are mutated in particular carcinoma, Warburg effect occurs continuously. So in this way, they basically attain their metabolites by diverging the glucose or by preventing the glucose entry into the oxidative phosphorylation, and they continue their needs by promoting glycolytic pathway. Next, altered metabolism is the autophagy. Autophagy is a basically process in which cell eats itself or self-eating. When the cell is nutrient deficient, it begins to eat its own organelles, and it fuses. For example, this was a cell here, and here is nucleus, and here is lysozyme, and here are its other organelles, and when there is deficient, this lysozyme comes and fuses with them and begin to eat and generate energy, which is used for the cell survival. However, the tumor cell also show an altered response to the autophagy. They can survive even bare energy demands. For example, if they are deficient in nutrients, still cancer cell can survive, and this survival is also due to alteration in their genetic setup. And depending upon signaling, autophagy can be a friend or foe of the tumor. For example, if a tumor is not receiving sufficient nutrition, and it is not even consulting to the process of autophagy, it continues to grow at its normal rate, what will happen? It will become nutrient deficient and will die upon. So, however, some tumor cells respond to the autophagy, and they become dormant in this way. And when they become dormant by adopting the autophagy, what happens? They don't respond to the chemotherapeutic drugs. It is because chemotherapeutic drugs basically target the rapidly dividing cells, and when a tumor cell becomes adopts autophagy and becomes dormant, they don't divide rapidly. As a result, chemotherapeutic drugs don't target them, and it leads to the therapeutic failures. Okay, so depending upon the signaling pathway and depending upon kind of mutation, autophagy can be a friend or foe of the tumor. Moreover, the cancer cell also show a special kind of metabolism, you can say oncometabolism, which can lead to the production of oncometabolites. One principal oncometabolite we will discuss here is the 2-hydroxyglutarate. 2-hydroxyglutarate. There is an enzyme called isocitrate dehydrogenase (IDH), which is involved in Krebs cycle, and it converts basically isocitrate to alpha ketoglutarate. So this is the enzyme which is involved in normal Krebs cycle, converting isocitrate to alpha ketoglutarate. However, in cancer cells or in cells which are going to be neoplastic, there is some mutation in isocitrate dehydrogenase enzyme, and when it gets mutated, then mutated isocitrate dehydrogenase enzyme, what it does, it converts alpha ketoglutarate to 2-hydroxyglutarate. Okay, so mutated isocitrate dehydrogenase convert alpha ketoglutarate to 2-hydroxyglutarate, and this 2-hydroxyglutarate inhibits enzyme of TET family, and principal enzyme inhibited is TET2 enzyme. You need not to go into the details of these enzymes. You should remember that this TET2 enzyme is basically involved in the regulation of methylation. It regulates the methylation of the DNA. However, when it is inhibited by 2-hydroxyglutarate, there is abnormal methylation of the DNA, and this abnormal methylation of DNA is not a change in the DNA sequence. Instead, it is a inherited change in DNA without changing the DNA sequence, and this change is basically a epigenetic mutation. So what is happening here? Isocitrate dehydrogenase got mutated. It converted alpha ketoglutarate to 2-hydroxyglutarate, which inhibited TET2 enzyme. As a result, there was abnormal methylation of DNA, and this abnormal methylation of DNA is an epigenetic change, and it can alter the expression of multiple genes. Gene expression is altered, and as a result, gene expression is altered. And as a result of this change, there is neoplastic transformation. So this entire process is called oncometabolism. This process is called oncometabolism. And this particular metabolite which was generated here was called oncometabolite. So in this way, the cancer cells alter their cellular metabolism, and this altered cellular metabolism can help them to survive. However, the altered metabolism can also lead to the neoplastic transformation by inducing the epigenetic mutation we studied here in the oncometabolism. Thank you.